Introduction and Basic Fish Genetics. Number 1. Explain the importance of a breeding program and its importance in aquaculture. The more important function and requirements of breeding programs are the increased production and product quality, increased productivity and cost efficiency, maintain genetic diversity breeds, support the conservation and use of specific breeds, and consider animal welfare and sustainable system. Aquaculture is the fastest growing food production industry, and the yes majority of aquaculture product are derived from Asia. The quality of aquaculture product directly consumed is now greater than that resulting from conventional fisheries. During the last 40 years, it has been shown that well-planned family-based breeding program yield high rates of genetic improvement for aquatic species. The genetics gains has been greater than 12% per generation for growth rates and for disease resistance when challenge tests are applied. Breeding programs are designed based on the productive cycle and behavior of fish and are integrated into the production cycle of companies to quickly incorporate the genetic improvement that comes from the breeding programs into farm production. Number 2, explain the principle of Hardy-Weinberg law. There are 5 basic Hardy-Weinberg law assumptions: no mutations, random matching, no gene flow, infinite population size, and no selections. If the assumptions are not met for a gene, the population may evolve for the gene. Genes' allele frequencies may change. The Hardy-Weinberg equilibrium is a principle stating that the genetic variation in a population will remain constant from one generation to the next in the absence of disturbing factor. The Hardy-Weinberg theorem characterizes the distribution of genotype frequencies in populations that are not evolving and is thus the fundamental null model for population genetic aims constants from one generation to the next in the absence of disturbing factors. A principle stating that both allele and genotypes frequencies in randomly matching population remain constants and remain in this equilibrium across generations unless a disturbing influence is introduced. Number 3, explain the function of all the parts of cell. Nuclear envelope, also known as the nuclear membrane, is made up of two lipids biliary membranes that in eukaryotic cells surrounds the nucleus, which enclose the genetic materials. Nucleus is the membrane enclosed organelle within a cell that contains the chromosomes. Nucleolus is the largest structures in the nucleus of eukaryotic cell. Lysosome, an organelle in the cytoplasm of eukaryotic cells, contain degradative enzymes enclosed in a membrane. The cytoskeleton organizes other constituent of the cell, maintains the cell shape, and is responsible for the locomotion of the cell itself and the movement of the various organelles within it. Microtubules. It is provide the rigid organized component of the cytoskeleton that give shape to many cells. Plasma membrane. It provides protection for a cell. The endoplasmic reticulum transport nutrition from one part of the cell to the other. Ribosome. It is the site of protein synthesis in the cell. Mitochondrion. It is generate most of the chemical energy needed to power the cell biochemical reaction. Basal body, it is motility, cell cycle progression, morphogenesis, and sensation. Flagellum is primary a motility organelle that enable movement and chemotaxis. Microvilli, it increase the cell surface area and thus facilitate the absorption of ingested food and water molecules. Vacuole, vacuole, it is handle waste product, and by handle mean take in waste product and also get rid of waste products. Centriole, it is organized microtubules that serve as the cell's skeletal system. Secretion granules, it is serve as storage pool for selected secretory products. Number 4. Explain the central dogma of molecular biology, protein synthesis. The central dogma of molecular biology states that DNA contains instructions for making a protein which are copied by RNA. RNA then use the instruction to make a protein, in short DNA protein, or DNA to RNA to protein. The central dogma of molecular biology is a theory stating that genetic information flow only in one direction from DNA to RNA to protein, or RNA directly to protein. The central dogma suggests that DNA contains the information needed to make all of our proteins, and that RNA as a messenger that carries this information to the ribosomes. The ribosomes serve as factories in the cell where the information is translated for a code into the functional product. Central dogma in Central dogma, an inheritance mechanism in molecular biology. Central dogma illustrate the flow of genetic information from DNA to RNA to protein. It is defined as process in which the information in DNA is converted into a functional product. Number 5. Explain the principle of mitosis and meiosis. Mitosis is the division of a cell into two daughter cells that are genetically identical to the parent cell. Mitosis is a process of nuclear division in eukaryotic cell that occurs when a parent cell divides to produce two identical daughter cells. Mitosis is conventionally divided into 5 stages known as prophase, prometophase, metaphase, anaphase, and telophase. Meiosis is the division of a germ cell into 4 sex cells, each with half the number of chromosomes of the parent cell. Meiosis is a process where a single cell divides twice to produce 4 cells containing half the original amount of genetic information. These cells are our sex cell, sperm in males, eggs in females. During meiosis, one cell divides twice to form 4 daughter cells. Meiosis is the type of cell division that creates eggs and sperm cell. Mitosis is a fundamental process for life. During mitosis, a cell duplicates all of its contents, including its chromosomes, and splits to form 2 identical daughter cells. Number 6. Cite an example of Mendelian inheritance in fishes and gene action in fishes. Inheritance is by units for particles now called genes which maintain their identity from generation to generation. It is also known as the law of segregation of genes. These units are present in duplicates in each individual. It is also called the law of independent assortment, which means that segregation at one locus does not influence segregation at another. Example of human autosomal medallion traits include albinism and Huntington's disease. Example of human X-linked traits include red-green color blindness and hemophilia. Gregor Mendel, through his work on pia plants, discovered that fundamental law of inheritance. He deduced that genes come in pair and are inherited as distinct units, one from each parent. Mendel tracked the segregation of pair, the ultimate goal of every selective breeding program is to improve the breeding value of the population. The breeding value is determined by the fish's genes. A farmer hope, hopes that when the improve the breeding value of his population will also improve its